Yeah, good morning, everyone. Welcome back to the second session of the third day of the FDP. Now we have Dr. Dipanjan Kosh, who will be giving a talk on role of CRISPR Cas technology in synthetic biology. So brief background, he has completed his MSc in 2000 from University of Kolkata in biochemistry, followed by his MTech in 2002 from IIT Kharagpur in the Department of Biotechnology, followed by his PhD from the same institute in the field of protein chemistry, cell biology, immunology, and cancer biology. Further, he has followed up with postdocs, one at University of Texas Health Science Center at Houston in the field of catalytic antibody and its engineering, followed by MD Anderson Cancer Center, Houston, in the field of lymphoma biology and genome editing. Then he returned back to India as a DST fast-track young scientist and at Calcutta University. Then he is currently working as an assistant professor in the Department of Natural Product at NIPER, Kolkata. Today he will be delivering a talk on role of CRISPR Cas in synthetic biology. Over to you, Dr. Dipanjan.
Thank you. Thank you, Dr. Shomashundram. Am I audible?
Yes, yes.
So, thank you, Dr. Shomashundram, for your kind introductions. So, I am Dr. Dipanjan Kosh, as you told. I'm an assistant professor in the Department of Natural Products. By training, I'm a biologist, synthetic biologist, or I'm a biotechnologist. So I work on the various part applications with the CRISPR Cas system, is modifications and in applications. So today I will talk about this application of CRISPR Cas system in synthetic biology. How this new technology, as you know, CRISPR Cas has become a household name. Everybody knows it, right? So because last year, two women scientists, Emmanuel Carpenter and Jennifer Doudna, they received Nobel Prize for their work of the CRISPR Cas. And from there, it has become a...
Dipanjan?
Yeah.
You are sharing your presentation or?
Oh, I'm sorry. Yeah. It's not, I forgot to share it. Thank you.
Yeah.
So it's as now, you can see my presentation, right?
Yes.
Okay. So as I was mentioning that last year in chemistry, two women scientists, they received Nobel Prize for in the recognition for their work of CRISPR Cas system. Okay. And the reason it is very important now, because CRISPR has become a regular techniques or tools for doing genome editing. So more specifically, very much specific genome editing, targeted genome editing. That part I will come in later. So this has become very easy, fast, efficient. Okay. So compared to other techniques that we used to use. So now here, the main thing is that, as we know, the CRISPR is basically a bacterial system for bacterial acquired immune system, and that has been used for eukaryotic genome editing for various purposes. Okay, one of the various purposes means you can use that for change your genes, tagging your genes, incorporate some mutations, revert some mutations, or you can use the help of that, you can introduce the foreign DNA and for their expressions. So here there are a lot of applications for CRISPR Cas system, and now new types of Cas also have come. So that we'll talk later. So since it is a biosimilar FDP on the biosimilar, so basically as my previous speakers have already introduced about the biosimilars. So basically, I just want to tell this basically biosimilars are nothing but the medical substances similar to the drug or therapeutic molecules. And the more importantly, how we the biotechnologists are involved in this, because the sources of biosimilars are the living organisms, which are basically engineered living organism, which are used by made by recombinant DNA technology. So we, being the uh, molecular biologists who are involved in this engineering process. So as you know, any biosimilars or any biologics, the product, there are two parts. One is the downstream processing, another is the upstream processing. All right. So upstream processing means how you can manipulate the gene, their expressions in various living organism systems. Okay. And thereafter, you have to optimize various conditions for getting the high expressions. Okay. You can get, your product can be either the cell itself or the ligand organism, the cell itself, or something, and something which is produced by these organisms. That is the engineered organisms. It can be intracellular, it can be extracellular. Okay. So whatever the biosimilars or biologics you see, they mostly come from the recombinant DNA technology. Okay, using the engineered organism, right? And you talk about the vaccine, you talk of the monoclonal antibody, you talk about the therapeutic peptides, or talk about the enzymes, or talk about the some recombinant proteins. All these biosimilars, those are produced by metabolic bioengineering through recombinant DNA technology. Okay, and now my point here will be how the CRISPR is being used for this type of productions of biosimilars. Okay, some products. Okay, as everybody already familiar with that, so since biosimilars has different names in different countries, and Europe it is called biosimilars, WHO calls similar biologics therapeutic products, in India it is called similar biologics, USA it is called follow-on biologics, so on, right? And every country has their regulatory authority to look into their how these productions of biosimilars and their efficacy and toxicity and everything. So here, as I mentioned, so coming to the biosimilars, which as I told you, these similar biosimilars are made, the biologics which are made inside the cell. Okay, so when you use a cell to produce your product, the cell become a factory. The new term has come up, it's called cell factory. Okay, where your cell that produces your product, and we the biotechnologist, we using the RDNA technology, we just engineer the cells with the facilitate them to produce the product you want. Okay, so that is the role of a biotechnologist in a upstream process. Okay, and then after your cell have been engineered, then your production level, you have to consider how we can make it more, and and then downstream processing, you have to consider how you can purify your product or other purposes, right? So this basically, you need to know a cell, how you can incorporate a foreign gene inside the cells and facilitate that expression, and in the run you have either what you want a protein, you want a RNA, or you want an enzyme, or you want a metabolites. Whatever you want, based on this, you need to optimize those things. Okay? You have to optimize the whole recombinant DNA process, and then you have to optimize the product, right? So that part I will come. I will focus on, this can be done in many ways, okay? Now I will focus on how the CRISPR-Cas is being used for these product formations or for the biosimilar formations, okay? Before that, since it is a heterogeneous population here, I'll go from very basics of things. Okay, so when you talk about any recombinant product, recombinant DNA expression, it can be your protein, recombinant protein, or RNA, or anything. So you have to think about the very basics of life. That is, your life, you have the DNA, which has all information, right? Encodes all your genes, gets transcribed, produce the mRNA, okay, or the messenger RNA, and these messenger RNA are eventually translated to give you a protein. So this DNA to RNA to protein, this is the two-step decoding process. Okay, so the protein which you want to produce, which are being produced in the cell, already this code is there inside their genome, correct? And that has to be expressed, and expressed to produce the product. So product, this is called, so product, if it is a protein product, it has to go through transcription followed by translation. It is a two-step decoding products. And in cell, most of the cases, the proteins are the doer of the cells. It can act as an enzyme. It can act as structural proteins. Okay? So basically, the DNA to RNA to protein, this flow is called central dogma of life. Okay, so central dogma of life, which is there in each cells. So basically, you have the DNA to DNA is called replications, then transcription happens to put the RNA, and then the RNA gets translated to protein. So this happens in every cells, and this is called central dogma of life. So basically, we take that thing, how using a gene, how you can express, you can do the transcription, and ultimately the translation using a cell, within a cell. This information you have to do. This thing you have to do to produce the product. Okay. So before that, as I know, so the gene concept was very long back, that in the classical principles of genetics were deduced by Mendel long back in 1865 on the basis of breeding experience of peas. And he assumed that each trait is determined by a pair of inherited factors that are now called gene. So he could see there is some factors that is coming from their parents to the offspring, right? So that factor, he visualized it, but he couldn't know what is its chemical nature. Okay, that factor is now, it's called gene. Okay, then Mendel's work was rediscovered in the 90s, and Hugo de Veris and others. So then in that time only, the gene, the William Johansen, he coined the term the gene. Ultimately, a new branch came called the genetics. More importantly, the paradigm was shifted for the gene when Rosalind Franklin took an X-ray diffraction photograph of the DNA crystals, and moreover, the people came to know that the molecule that is responsible for the gene, making your gene is called deoxyribonucleic acid. And then people solved the structures, and in 1950s, Watson and Crick, they built the first model of DNA based on the Rosalind Francis X-ray diffractography, and they say now everybody knows this is the double helix structures. As it is known, I'm not just giving this because this is the one of the discoveries that gave the science the paradigm shift of the scientific achievements. Okay, that took from one level to another level. Okay, so then another important discoveries is came from the restriction enzymes. So in 1962, Werner Arber, a Swiss biochemist, provided the first evidence for the existence of molecular scissors that could cut the DNA. And he showed that E. coli bacteria have an enzymatic immune system that recognizes and destroys the foreign DNA and modifies the native DNA to prevent self-destruction. So, at the two things he mentioned, that one, these some molecules that acts as a molecular scissor, and it protects the host. It protects the bacteria, okay, and acts as a bacterial immune system. Okay, so the molecular scissors that cut the double-strand DNA molecule at a specific point and found naturally in one variety of prokaryotes, mostly in bacteria, and important tools for manipulating DNA. So rather, because these are why does bacteria produce the restriction enzyme? Because bacteria wants to destroy the phages, okay, bacteriophages or the foreign DNAs that invade or attacks the bacteria. Okay, it's a defense mechanism of the bacteria. Okay, and that defense mechanism now being used for doing the recombinant DNA technology, and it has been well taken. Okay, so basically the restriction enzyme they are called because they are restricted for their activities. They can bind a particular sequences, and they can then you can make the cleavage. Okay, so either they can make a blunted type cleavage or they can have a sticky end type cleavage. But they can only bind to a particular sequences. Mostly they are palindromic in nature. Not always, but type 2 they bind in the palindromic sequences. So the reason I am telling about the restriction enzyme is, so they have a two part. One is the endonuclease part, another is the DNA binding part. Okay, so when you use the DNA binding part, so DNA binding part, as you can easily understand, this part is used for binding to the DNA sequences, right? There are some motifs are there like zinc finger motifs and others. Those are there too for many transcription factors, because transcription factors they bind to the DNA. So like these type of DNA binding motifs also these type of restriction enzyme they have. When they bind to a DNA sequences, as I mentioned, they are very restricted because they can bind to a particular type of sequences. Then only these endonucleases attached to this, they can cleave it. Okay, so they can be programmable. That means you can take a DNA binding domain of one restriction enzyme and endonuclease the other restriction enzyme. You can still get a cleavage. So you can play with this, right? You can program these DNA binding domains with the nucleases. Okay, so you can make your own type of things, changing the new DNA binding domain and the nuclease domain. These two domains, after giving the permutation combination, you can make a programmable endonucleases. Okay, so now there are people used, when they were using the programmable, there are people use other DNA binding motifs. Okay, and there are many DNA binding proteins, as I mentioned, the zinc finger proteins are one of the DNA binding proteins. Okay, so then what they did, they one zinc finger motif can bind to a triplet. Okay, so if you have an array of zinc finger motifs, then they can bind to the 9 nucleotides, because one gene finger motif can bind to the 3 nucleotides. So in this way, you can make a gene finger proteins array of you can make a gene finger proteins with combinations, so that you can target your target sequences, right? So these are what we are making. We are trying to make very targetable, very specific, or very sequence specific DNA motif binding proteins that can bind to a particular sequence, so that if you attach the endonucleases, they can cleave it. So in case of gene finger nucleases, so people use the array of gene finger gene finger proteins, okay, one which carries 3 or 4 gene fingers motifs, so that they can bind 9 or 12 target site in one stand. So you can use a 2-stand targeting. So one array you can use for targeting the upper stand, another you can use for the lower stand. And both are cases, the FOC1 enzymes are there such a way, so two part, when they bind together, then the FOC1 enzyme can act and cleave. So this is the basically how the gene finger nucleus acts. So basically it is DNA protein dependent DNA binding followed by the cleavage. Okay, so basically you can manipulate, you can program these gene finger proteins to bind a stretches of DNA sequences that you want to manipulate. Okay, so in the other cases, the TAL proteins, it is a transcription activator like and enzyme like proteins. These proteins, there are 4-5 types of proteins. Say the proteins are all of same sequence except these types of RVD, which is very specific for binding to a particular nucleotide. So here in this case, for one nucleotide, you will have one type of TAL proteins. The second nucleotide will have a second type of TAL proteins. So in this case, you can make a array of TAL proteins, okay, and then you can attach the nuclease so that it can cleave a particular sequences. So it is called TALEN. Okay, so first came the gene finger protein, which in both cases, and then came the TALEN. So both cases, so basically DNA binding part is a protein, and then these are attached. These are what you do. You design the DNA binding things by adding or various. You can program that using various DNA binding proteins together. Okay, one after another. Okay, either gene protein or TAL proteins. Then those proteins are attached with the endonucleases that can cleave a particular crystal. So here, both cases, gene finger and gene finger nucleases and TALEN, both cases, the protein-mediated DNA recognitions happens. And the problem is that making a protein, a specific protein array of these proteins is difficult, very expensive, and very much difficult, and sometimes the recognition is also difficult. And so making a successful gene finger is, you have to play with many combinations and others, then you can get up a successful one. And their efficacy is not that good, but specificity is also there. Okay, so TAL protein is even bigger, so it's more complex, right? And getting that big protein expression and all these things is a challenge, but still they have a better efficacy, okay, compared to the gene finger. But when the CRISPR came, the advantage of CRISPR is it is basically RNA mediated genome editing. So when it is an RNA thing, so what you have to do, you have to get a RNA, you have to express a RNA, and with the specific sequence where you want to do the manipulations. So that is rather easy than you have to making a proteins and array of proteins to getting together, and that is more complex. Okay, so in this case, I'll talk how CRISPR works, but you just remember the difference between the other DNA targeted DNA technologies to CRISPR is that CRISPR is the RNA mediated genome editing, and which requires a RNA, or another thing is that the endonucleases. Two part it requires. So I'll come to in details about that as I go on. So because as I already mentioned, when this CRISPR was introduced, and as it is RNA mediated and easy to make, so its price will be less. It's high efficacy, accuracy, and it's simple, easy to make, very less time consuming, its price is down, so it's having a very good accuracy. So it became a game changer. Okay, so evolve the field, this field evolved a very faster way. Now you can make any knockout, any modifications with this CRISPR Cas system with ease. Okay, that's another what will the paradigm shift, I will say, that this system which came and in this type of genome editing field has gained is momentum. Okay, now you can target anything, you can play with it, right? And you can cut, paste, and this type of engineering you can people are trying to do, make your all type of things. Okay, so now going back to some of the history of how CRISPR came. Okay, so basically the first evidence of CRISPR came from this paper, JVC paper in 1989, sorry, from the Japanese group. Okay, Kozo, Makino, the Japanese group. What they found the one K12 strain of the E. coli. In the genome, they found there is a repeat special. There is, so they found there are 29 base pair. Okay, conserved sequence. Okay, the 29 base pair conserved sequences. Okay, the highly conserved sequence of 29 base pairs containing the inverted repeats of 7 pairs that appears 14 times. So this is as a conserved sequences, okay, having the inverted repeats. So you see the citers, these are say GG, this is CC, TAT. So this is their terminals are inverted repeats. These are the complementary to each other. So this is called inverted repeats. Okay. So there are how many terms? There are 14 terms repeat, and they come after a particular 32 or 33 base pair apart. So you have a conserved repeat sequence, okay, in the genome, okay, and that has only 14 terms, and they are after a particular apart of 22 or 23 base pair. Okay, so this type of repeat sequence, why there will be in the genome, but this is finding give the first startup. Okay, this is the first thing that started from where the whole concept came. So they reported it. So in the genome, you see these type of repeat sequences are there. Okay, but what is their role and why they are there? That they couldn't understand at that point. Later, lot of bacteria, archaea, the reports came. They also have that type of, because after that the whole genome sequencing was rather easier. Okay, for these prokaryotes and others, and that gained the momentum at that time. So getting the more and more information about the genome sequences of other prokaryotes, archaeas, and others. So that's become very easy. Okay, then lot of information came. So not only this may be the last paper, this may be the first report, but other reports also came from various bacteria like E. coli and others, archaeas, they have this type of repeat sequences in the genome, and they're clustered. They're not randomly spaced. There is a clustered leaf space. So the CRISPR term was coined. Okay, this type is very common in the genome. So the terminology was called CRISPR. That is, the full name is clustered regularly interspaced short palindromic repeats. Okay, this was coined by the these Dutch people. Okay, in 2002. Let's see how the CRISPR locus look like. General CRISPR locus look like in the genome. Okay, when you see in the CRISPR, there I told they are clustered in a part of the genome. So how they look like? So CRISPR is locus look like this, where you have you can see the black diamond. These are the repeat sequences. Okay, so there will be a lot of repeat. In between the repeats, there are the sequences called spacer sequence. So you have a repeat spacer sequences. So repeat spacer are your inverted repeats, those are the conserved sequences. But the spacer sequences are random sequences. They are not the conserved sequences. Okay, but their number may be very close to same, but they are not the same sequences. Okay, so how many times repeat can be there? We can have 2 to 350, right? That repeats per locus you can see. Okay, and so they are mostly invariable sequences. Okay, and per sequence, repeat sequence itself will be 21 to 48 base pairs. On the other hand, the spacer will be 1 to around 374, 350. Okay, and these spacer sequences, unlike the repeat sequences, the spacer sequences are variable sequences. Okay, and these can be 26 to 70 base pair long. Okay, most importantly, I'll come to this. Adjacent to this CRISPR locus array, there are some genes which have some role to play in the expression of the CRISPR system. Okay, these are called CRISPR associated gene or Cas. There are many Cas proteins are involved for processing of this RNA, CRISPR RNAs, and then making a final CRISPR RNA which actually acts. I'll talk to that. And more importantly, the spacer sequences have found that they are the foreign DNA. These sequences are, so some sequences have been found in the viruses and foreign DNA sequence. So here's a clue. This is your indigenous sequences. These are foreign sequences. Why these foreign sequences will be there in the genome? That's a clue. Why that should be there in the genome? You should have a sequence that has a similarity to the foreign DNA. So these are actually the memories of the foreign DNAs. Okay. Then the scientists thought about it, and then it may realize that it has something to its immunity. Okay, just like a memory cell. So how did the scientists find out about this immune system of bacteria? So in 2007, okay, Barango and his team published a science journal, published a paper in Science Journal, an experiment in which they used a common bacteria, Streptococcus thermophilus, as their model. The bacteria is found in the curd. They observed that after a virus attack, new spacer were incorporated in the CRISPR region. Okay, so very nice observation. So they found that the virus attack, the new spacer has been incorporated in the CRISPR region. Moreover, the DNA sequence of this spacer was identical to parts of the viral genome. So the virus infecting the DNA, and there's some sequences are incorporated in the CRISPR locus, and this sequences is very similar to the virus. It came from the virus, that is obvious. They also manipulated the spacer by taking them out, putting a new viral DNA sequences. Okay, so how they manipulate? So they use the various viruses, then saw the various sequences. Okay, in this way they were able to alter the bacterial resistance to the attack of the specific virus. So what they did, if you can put a particular sequences in that genome, okay, and then they infect the viruses, and they were seeing that bacteria was resistant to the virus infection. Thus, the researchers confirmed that the CRISPRs play a role in regulating the bacterial immunity. Okay, so now the paper came, another paper, these are the paper from the Jennifer Dunda and Carpenter, who were the instrumental in that particular bacterial adaptive immunity things. So their group also found that programmed dual RNA guide RNA encodes in adaptive bacterial immunity, and that's how it works. As I mentioned, the work is very good. Where this is what you see is that CRISPR array in the genome. This is a CRISPR locus, and this is a CRISPR array. Okay, this is CRISPR associated genes are here. Now, whenever the plasmid DNA or bacteriophage DNA come, they are incorporated in the CRISPR array. Okay, so they can have it is called spaces in the spacer sequences. Okay, so the spacer sequences are incorporated in the genome. Okay, and then whole things are expressed as a pre-CRISPR RNA, and the Cas proteins, they cleave them and chop them and produce a active CRISPR RNA, which along with some Cas proteins, they bind with the CRISPR RNA, and then they interference and they cleave these. Whatever the next time that this virus DNA comes, they can recognize, because these sequences are already there and they are being expressed as a CRISPR RNA. And this CRISPR RNA, along with this Cas protein, they can bind the same sequences when they come, and they can cleave it. Okay. So the sequence that comes from the foreign DNA is called protospacer, and these protospacer sequences, after these protospacer sequences, there are some motif or the DNA sequences. Those are very important for this Cas protein binding or the CRISPR RNA binding and Cas protein activity. This is called protospacer adjacent motif. All right, there are each Cas protein has its own particular PAM specificity. What is PAM? Protospacer adjacent motif sequences. Okay, so various type of CRISPR systems are there, and their name is also there. So and you know that type 2 is being used for particular genome modifications. Okay. So whatever I told, that starting from in 1987, from the Japanese group, they found that 29 nucleotide tandem interval repeat sequence in E. coli, that was reported 14 times. And then in 2002, the CRISPR name was coined. 2005, the spatial sequence of bacteria were found highly homologous with their viruses. Okay, and then in again, just 2010, genome editing techniques of CRISPR Cas were developed in 2012 for the specificity, and in 2015 or 16, these were used for inactive the oncogenic mutations. CRISPR. So they were largely after 2012 to 2016, 17, lot of genome editing work was done, and not only that, knockout mice was made with the CRISPRs, all the not only in human, all the mouse, and even in the plant system, in zebrafish, and everywhere, the CRISPR is being used in protozoa and everywhere. Okay, and then CRISPR, some problem in CRISPR has been now recognized. Okay, there are more types of CRISPR Cas systems are now coming up, and in 2020 the Nobel Prize has come, but still this CRISPR system is evolving. Okay, Now, as I mentioned you, that a type 2, there are three types of CRISPRs when they are produced, type 1, type 2, type 3. You did not go in details. And just remember, when the type 2 is being used for genome manipulations. So what is the type 2? In this type 2, when you have pre-CRISPR RNA is produced, all right, after, and there is a trans-acting RNA is also there, okay, along with the Cas gene. And the main important, the endonuclease here is the Cas9. Okay, the other Cas genes are there for the cleavage of this RNA and making the mature CRISPR RNA. Okay, so when the mature CRISPR RNA is produced, okay, that has some homology with this tracker RNA, okay, complementarity, and the tracker RNA and the transacting RNA and matured CRISPR, they have the homology. They make this type of two things, mature CRISPR and processed tracker RNA. They bind to each other, and then they can bind to the Cas9 protein. Okay, so first thing is the tracker RNA and the matured CRISPR RNA. They bind each other, okay, and then they can bind to the Cas9 protein. Okay, then they dock it. So ultimately, the first 20 nucleotide, this mature CRISPR RNA has the particular sequence of the target DNA. So then it can dock the whole complexes to the particular target DNA. And then this Cas9, which is basically endonucleases, it can cleave it. Okay, this is how the bacterial adaptive immunity works. Okay, it is how, where from you get it. Now, life has become easy. Previously, what you have to use the two RNA, right? Host tracker RNA and the CRISPR RNA. Instead of that, can you make a synthetic single RNA? Yes, people have done it. They made they have stapled these two together into a synthetic one RNA. It's called gRNA or the guide RNA. Instead of two RNA in the type 2 CRISPR system, which are the transacting RNA and the host CRISPR RNA, you can now make a synthetic one, single nucleotide one, which is basically 105 nucleotide long. Okay, this is a single one. Basically, you can make them instead of two RNA, you can make a single RNA. Okay, life has become easy. So you have to have a two components now. Okay, one is the gRNA, another is the Cas9. And now, as I mentioned, that this gRNA, the first 20 nucleotide is the target DNA sequence. So your life, your job is to change these 20 nucleotide sequences, target sequences, to where you want to target it. So you can design it, your target sequence, and then you can manipulate the gRNA sequences with these target sequences, so that you can dock all these complexes to a particular sequences, right? So basically, the first 20 nucleotide, which is the target sequences, you can change it. So you have to have a system where you can clone these 20 nucleotide only, and the whole backbone will be there. And so basically, what you need two things. Okay, one is that basically the guide RNA, as I mentioned. The first 20, this is the single nucleotide, 105 nucleotide longs, a single stranded, and the first 20 nucleotide is the target complementary region, and then a protein is called Cas protein. When they make a complex, and this complex can bind to the particular target DNA, which is the complementary of this particular sequences, right? And then the endonuclease can cleave it. All right. So what you have to do, and then so you have to produce a gRNA, and you have to produce a Cas9 in a cell, where your gRNA you can engineer with a particular sequence where it will bind, and the whole complex will bind there, and the Cas9 will cleave it. All right. So how does the when it cleaves it, what happens? So basically, what you do, you make a gRNA and Cas9 express in the cells. They are such a way, then they cleave a double strand break, and that double strand break can be repaired by pathway called NHEG, non-homologous end joining, and another pathway is called homology dependent pathway. Okay, and when it happens, the NHEG wins the race, and NHEG makes there some types of changes, insertion or deletions. Okay, if you do the insertion or deletions, so there will be some sequence will be deleted or inserted, the frame will be shifted, and then you don't see any protein expression. Protein of is not expressed, so your gene is knocked out, and it's a permanent one. Or you can use the donor based, homology based, you can insert it, okay, where you can insert a gene, you can insert a stop codon, whatever you want to do, manipulate it. Okay, that's very much important. The HR based double strand break repair is the very important for synthetic biology or any type of applications. Okay, for doing the knockout, just simple you can rely on NHA, EJ, or for getting some gene tagging or inserting some the particular type of mutations or reverting mutations in either case, or inserting a gene, you have to rely on HR. Okay, so people have used the multiplex, two papers, science paper came back to back paper in class, the RNA guided genome editing engineering by Cas9 and this CRISPR Cas system. These two papers came in a same science paper with these two science editions, science volumes, two back to back paper came with the using the CRISPR. People have done the targeted genome editing. Okay, and basically what I mentioned is that you need a single RNA, the endonuclease Cas9, they bind, make a complex, that complex to go bind to the sequence with the PAM adjacent protospecial adjacent motif. This is the particular red portion is the sequence complementary with this CRISPR sequence, first 20 nucleotide, then they make a double strand break, Cas9 makes a double strand break, and then indel happens, non-homologous end joining, so insertion deletion happens, frame shift happens, and you see the knockout. Other way, you can do the homology dependent. You have to give a homology arm, and that homology arm can be incorporated. You can insert any sequence you want. Okay. So this is the very easy system. This is the way the CRISPR works, but CRISPR was never out of the controversy. Okay, the first controversy was pivotal the patent. So as you know, there was a lot of court lot of debate regarding the patent, and lot of court battle was there, and then the CRISPR patent was given to the Board Institute, okay, after a long legal battle. And then the things you have heard about it is that CRISPR babies in China is a scandal, growing scandal, where a Chinese doctor has used the CRISPR Cas technology to make a test tube baby where from the sperm and ova from HIV infected parents, and he removed the he cured the HIV, and the baby was born without HIV. So this is claiming that you can use CRISPR and you can remove the gene you want, but still you cannot, because the CRISPR, it became a scandal because it is not ethically correct, or it is not allowed for the therapeutic purposes, still for the human uses. Okay, so why? Because this is not a foolproof technology, because it is high efficiency off target effects are there. So it can tolerate some of the mismatches. So there is off target efficacies, off target effects are there, and that gives its limitations to be used as a therapeutic purpose. Okay. So this limitation, as I mentioned, so how people used to get rid of the limitation? What are the ways? There are many ways. People are trying to engineer the Cas proteins. People are trying to engineer the gRNA. People are trying to use the various nucleotides. All right. So all these things are, and people have changed the Cas protein also with other Cas systems. Okay, these are the way people are trying to get rid of the these limitations, the off-target effect. Okay, so one of the effect we have used is that using the paired nicases. So now, what is nicase? Okay, so nicase is basically, if you see the Cas9 protein, it has multiple domain. Okay, so one of the domain, two domains is called RACPC and HNS. These two domains are present in the, one is the N terminal, another in the C terminal. Okay, so two mutations, if you do any of the two mutations in the one of the domain, is a D10A in the N terminal and H840A in the C terminal, either of these mutation, what happens? Then it can, instead of making a double strand break, the enzyme can cleave one of the strands. Just the, okay, it can nick one of the strand, not the double strand breaking. So basically, that if you have any of these mutation that in the Cas protein, that is called nicase. Instead of the wild type Cas9, you can have the nicase type of things. So if you use a paired nicases that which are targeted to one in the upper strand and another lower strand, so ultimately you are making a double strand staggered, long staggered double strand break. Okay, you can have to give this target site around 40 to 50. And what is the advantage? So how the nicase help in this get rid of these limitations, to overcome the limitations? So basically, nick repair is a safer one. If there is a off-target nicking, so it will be just be repaired. There should not be any mutations or insertion deletions type of things. So it is safer. Even there is a very less of chances that two off-target sites will be very close to each other. These are rare opportunities, so that you can have a two off-target cleavage nicking very close to each other. Okay, but so people use these pair nicases, but disadvantages is that you have to use two pairs of nicase. You have two target sites at the time. Okay, and they're very should be 40 nucleotides apart, but their efficacy is not at is okay, but we used it and we compared it with this. So what we did, we used the paired nicases and saw how it is used for knocking. Okay, we are more important, instead of doing a knockout experiment, we are more important doing a knock-in experiment. So here what we did, we knocked out a CXCR4 gene, okay, in JAK1 cell line, and where you see that there are two target sites and two nicases have been targeting these two sites. And you see that these are the knockout populations, and almost 50% knockout you can achieve. At the same time, so we wanted to use introduce a GFP in the same locus, and you see that their expression of GFP is also there. So in the knockout population, though the ratio is NHEJ is very high compared to HR as expected, but thing is that pair nicase is working and working okay, acceptable is efficacy, and this is much safer. So these are the one thing that we are using, and this could be one process people also are using for doing the genome editing of the, but still not accepted for the therapeutic purposes. All right. So we used another one, the how to improve these knocking populations. So we use some small inhibitors, okay, to block the NHEJ. And you see that if you use a combinations of various inhibitors, you can increase the HR populations. Okay, homologous dependent knock-in is increased. In the genome also we have seen this. SCR is one of the inhibitors that works in the that blocks the DNA pole, right? And that DNA ligase 4, that is one of the NHEJ pathway. It is a part of the NHEJ process, and then you can see with the increase of the SCR7 and even using the pair nicases, you can increase the percentage of knock-in in the genome. So then this is the basic how you can use the CRISPR Cas, how you can modify the CRISPR Cas, and you can use for the knocking processes. Now we are more we want to use that for the production of the small molecules or production of the synthetic biology for the production of therapeutically important molecules. So how we can use this for targeted knocking of the genes and expressions of them, and then their productions. Okay, so this is the first paper, that production of the antiviral drug precursor artemisins, that artemisinin in the engineered works. So that is the pioneer paper in the field of synthetic biology with the engineered gene yeast, giving the synthetic biology or metabolic bioengineering modifications. So gives us a confidence, yes, we can do it, we can make these molecules, or you can make the lot of therapeutics, biologics using the CRISPR systems, making the engineering the yeast or the bacterial systems. We wanted to do a metabolic bioengineering glucocalin to produce the glucocalin, which is a natural compound from the gallic acid and beta-D-glucose. It is there in the amla and oaks. This is how it looks like. So this is the glucocalin structure. It is found in the amla. It has a lot of pharmaceutically important compound like this. It acts as an antioxidant. It has been reported to protect the hepatic carcinoma. It has been used. It has been shown to predict anti-inflammatory. Okay, so too many reports are there. Okay, so for glucocalin and even for the anti-diabetic also, people have their reports are there. So this is an important natural products molecules. So, but getting from the amla is not a very economically is not very suitable. So what we wanted to do, can we make it metabolic through the metabolic bioengineering? So using a CRISPR Cas system, as I mentioned here, in the yeast system, so all the vectors are available. Now here in this PML 107, what you can do, this vector is equipped with the Cas9, as well as there is a portion where you can clone your target site just before the tRNA backbone. Okay, and then we used to make it so we targeted a TRP gene and wanted to produce the enzyme UDP glucosyl transferase, which is involved for the formation of glucocalin from gallic acid and UDP glucose. So just we want to express this in frame of a gene, TRP1 gene, and then we want to see the expression of the glucocalin. So that was the one step thing, so that your gene will be expressed, incorporated, that will be expressed in frame of one of the gene, and then that particular foreign enzyme that will be expressed, and that you can, if you give the gallic acid and ADP glucose, then you can check the formation of glucocalin or not. That we have done. Before that, we have seen we can modify this type of yeast producing their both GFP, RFP all together in the same locus, and there you can see the expression is there, and the same gene has been produced, and then you can see the expression of glucocalin using this engineered yeast. So we have to optimize, we are doing that, optimize the high expression, that part is going on, but glucocalin production has been confirmed. Here another work what we're doing here is that probiotics modification, improvement of the probiotic strain using one strain using the BSH gene, which basically bile salt hydrolyzes the bile salt to unconjugated bile acid they precipitate, and then you can use that for improvement of one strain is called Polar D. Okay, this Polar D is being used as a probiotic. So we want to use this BSH gene from another probiotic strain. So make this this strain improved strain, make an improved strain, so that this probiotic already is being used for as in the probiotic strain, the polarity is used in the probiotic, so it has a improved strain we can use with a food grade, or okay, that will we can use for the for the lowering of the cholesterol. Hello. Okay, so so this is the way we are doing already. So basically, the application of CRISPR Cas in metabolic bioengineering. So there are a lot of application people have done it. Okay, so people have made the lactic acid, this incorporating of lactate deaminase gene for the ethanol productions. Okay, they're making the 4-point deletion mutations, or they have used the nalingening, one of the component of the what is called the important natural products. Okay, so there are many things, some biofuels they have used. Okay, riboflavin people are produced. Okay, not only in the classical yeast like E. coli or yeast like Saccharomyces cerevisiae or E. coli, even non Rhodocappas on others they have used, strains they have used, and they see they constantly in a fermentation people are making those productions, and commercially these engineered these host or microbes are producing the products commercially. So this is a successful applications where people are using for productions of commercially importance of the molecules, small molecules. All right. So there are a lot of, this is a few I'm just telling, there are a lot of examples people this use. Okay. And this is another important one I'm showing the how people use for production of amino acid like L-valine. All right. So this is L-valine is produced when they actually this pathway is both the used for the isoleucine and valine, but the enzyme that has been used for initial part is the AHAS. So this enzyme has a two substrate. One is the pyruvate, another alpha ketoglutarate. Okay, so what is happening? So then affinity is here higher. So this always take the isoleucine production is high rather than L-valine. So what the engineering people have done, whatever you see the dot blue color, there you see the gene has been knocked out, okay, or knocked down. Okay, most of the cases knocked down. All right. So, and whatever you see the red one, that means they have overexpressed, or they have forcibly they have given the some facility to that they can go to that way. Okay. So pentose phosphate pathway has been the forced glucose 6 phosphate has been forced to go to the pentose phosphate pathway, and then it produces the pyruvate. So you want a starting material of pyruvate high, so you have to force that to produce this pyruvate more, And at the same time, the downstream of the pyruvate, so make sure the other things cannot use, only it is being used by the enzymes HAS. And as I already mentioned, the affinity of the enzyme for these 2-ketobutyrate is higher. So you have to knock this ketobutyrate, your expression should be down. So therefore, you also knock down some of the genes so that the ketobutyrate is going down. So in that way, you can forcing these whole things to go to produce the L-valine. All right. So there are a lot of strains have been produced, and now almost 200 times higher expression of L-valine. You can get the expression up to the 2, if I am not correct, 2 millimolar per liter production of L-valine is produced. Okay, this is very much successfully produced. Okay, and now the CRISPR, they are being used now doing some treatment or the epigenetic modifications of the some of the genes, and it has been used for the cancer and other detections and modifications. So what you do, so basically the histone and the epigenetic modification, you know that there are some histone methylations that there or DNA methylations that there. So what they do, you can when you can block them, okay, Dicasnan fuse with KRAB and DNMT3 tags, which leads to down regulation of the gene. Okay, so whenever the acetylation is there, okay, in acetylate condition, the proteins, the DNA, they are expressed high. So if you use the deacetylation, if you do, you can down regulate it, right? Or you can sometimes some methylations also, you can remove that demethylations, or you can make some methylation, you can incorporate some methylations, so you can also down regulate. So what you do, you use this gene which actually methylates the DNA, all right, or the histone. That how you do it, you tag your Cas9 or Dcas9 to that Cas9, so that you target to particular sequences, and nearby these tagged methylated methylases can methylate your DNA or the histones, and they can down-regulate the gene expressions. On the other hand, you can use the activate some of the gene expression, okay, by acetylations. So you use the enzymes for histone acetylations, and then the expression level will be higher. Or they can use another way, some of the transcription activation factor binding site, okay, people use, and they can incorporate a lot of transcription activator, and the promoter expression level goes up. Okay, these are the way people use the CRISPR for epigenetic modification and as well as the high expression. Okay, and this another way is that the RNA aptamer and others tagging you can do for, as I mentioned, for incorporating is the transcription factors and gene expression, higher gene expressions. Okay, so another is that use of CRISPR for the CAR T cell engineering. Okay, so after the CAR T cells, as you know, this is the application of CAR T cells for the to kill the cancer, because you are engineering the T cells so that it can attack the particular the cancer cell which are expressing some of the antigen. So basically the VL and VH domain is the antibody sequences for the particular cancer specific antigens. Okay, and you engineered that in your T cell receptors. Okay, so your T cells receptors are expressing this particular VL and VH specific to the cancer specific antigen. So now these type of CAR T cells, when you introduce into the patient, because you take the patient T cells, okay, you collect them, and then you engineer them, make them high concentration, and then give it back to the patient, so they can easily lyse the cancer cell, attack the cancer cell. The T cells are now targeted T cells. They chimeric antigen receptors, the CAR means. So they have, you made the chimeric receptors by engineering them, and this in these cases, getting them, making them, you can make it very easily by using the CRISPR system, or getting them incorporations into the, and after the gene is made, you can incorporate in a T cells in the targeted way by CRISPR. So the whole CAR T gene, this receptor, chimeric receptor gene, okay, and this can be engineered, and then after this, it can be incorporated in targeted position in the T cell genome by CRISPR, and these are being used. Okay, and it's a another one that saves lives. A lot of people, they therapeutically they can use the CAR T cells, and it is another the game changer in the cancer treatment. Now you have recently you have heard about the CRISPR is being used for detections of the uh, detections of detections of the various viruses, like the coronaviruses, and this Fan Jing had made one detector, which is he named after the great detectives Sherlock Holmes, the detective characters. So he called it Sherlock. Okay, and this is basically he used, instead of Cas9, he used the Cas13 things, which can detect the RNA, and it can, after getting the particular sequences, it randomly getting the activated, and then one of the, he has a cleavage reporter which can be cleaved. He has a reporter which can be cleaved, and you see the signal coming. So basically, they are using the signal reporter which with some fluorescent and others. So after the activations of this particular, when they see the Cas specific RNA, the Cas13 gets activated, and it gets randomly cleaves this reporter, and you can see the expressions of these reporters, okay, and see that can easily quickly detect it. So within 5-10 minutes, you can easily see the detectors. Detect those expression viral presence or not. Okay, so in India also we got the Feluda. Basically, Feluda is not but Cas13, it's a Cas9 base, but it has been used many times. You heard in 2020 October November, and from the IGIB, two scientists from IGIB, they used, and they already found that this, yes, we based on that same purpose, we can use the Cas9, and they use the therapeutic purpose, similar detection purposes. Okay, so they named the Indian this whole kit as after the name of the Feluda, the famous detective character by Satyajit Ray. Okay, so these are the use only I'm showing, not only for the therapeutic purposes of production of this biosimilar molecules, also for the detection of the various things. Now CRISPR is being used. So basically, what I told is that what advantage and CRISPR can provide. Yes. So previously, because the now pre-CRISPR era and the post-CRISPR era, the improvement of existing therapy like the CAR T cells, target drug targets, help find the drug targets, and help drug testing candidates, getting the cell lines available with modifications, making these your knockout mice easy. Okay, so all types of modification has become very easy. Very for testing purposes, it has no problem. but therapeutic purposes, it is not yet ready. Okay, but for using the CRISPR system for production of the small molecules or production of the biosimilars, biomolecules, you can use and you can modify the cells, prokaryotic cells or yeast cells, or even you can use this for eukaryotic cell modifications, right? But for the therapeutic purpose, CRISPR is not yet ready, and that's an open challenge now. One can, if you can make this safer, okay, for therapeutic purposes, now you see the whole world will be after you. Okay, with this note, I thank you for your attention, and I'll be happy to answer any question if you have.
Yeah, thank you, Dr. Dipanjan, for introducing this CRISPR Cas system in synthetic